In this lecture, we will discuss about protein secondary structures. In the previous lectures, we discussed about the primary structure of protein and what are the various information we can obtain from the amino acid sequences. We discussed about various features or properties, right. We discussed about amino acid occurrence, composition, molecular weight, average properties, hydrophobicity profiles, right, and how to construct profiles, and then how to align the sequences, right. We discussed about the different aligning methods, right, how to identify a sequence which is closely related to your own sequence, right. So, and various other parameters. In the last class, what did we discuss?
Non-redundant.
Yeah, because if you want to analyze a large scale data, if you have large number of sequences for example, then if you include all the sequences it may introduce a bias. So, we discussed about the redundancy of the sequences and how to eliminate the redundancy from any data set, right, with an example of amino acid sequences, right. So, how to construct non-redundant data sets, what are the different software we discussed in the class? CD hit, BLASTCLUST and PICES, right. What is the mechanism or what is the principle used in CD hit? K means clustering, right. They use a clustering technique, right, to obtain non-redundant sequences. Also, they instead of doing the complete sequence alignment, they try to construct the short peptides of different lengths and see how many residue segments are present in different sequences, right. Based on that, they developed an algorithm to obtain non-redundant sequences. So, what are the advantages of having CD hit?
Large amount of
you can handle large amount of data, so it is very fast, right. You can see it is reliable resource. Then when we have the disadvantages, so what are the disadvantages of CD hit?
Not suitable for very low identity.
Yeah, it is not suitable for low identity. For example, if you want to have the sequences of 20% sequence identity, right, that is not possible with CD hit because it uses up to 30% or 40%, right. Then the BLASTCLUST, you can get standalone version from the BLAST site, right. Here they can handle different sequence identities. Then we discussed about another server called PICES, right. So, here also you can get the non-redundant data set, but here the limitation is it can handle only limited number of sequences. So, we discussed several aspects and several information you can get from the amino acid sequences. And today we will discuss about protein secondary structures, right. So, what do you mean by secondary structure? Because going from the primary structure to secondary structure, it is one level up. So, we have more information than we get from the primary structure amino acid sequence. So, then defining secondary structure, how can we define secondary structures?
Periodic arrangement.
It is periodic arrangement, right. It is regular and the periodic arrangement of amino acid residues in a polypeptide chain, right. So, mainly these arrangements are maintained by hydrogen bonds between some specific atoms. Right. If you see the regular secondary structures, the hydrogen bonds formed between the amide hydrogen, right, NH, and the carbon and oxygen, right, CO, of the peptide backbone. So, if we discuss about any secondary structures, it is mainly formed by the residues which are in the main chain, not in the side chain. This is what we define the different secondary structures. So, based on the regular arrangement of these residues, there are different types of secondary structures. So, what are the commonly occurring secondary structures in proteins? So, alpha helices, right, because the first they identified the helices in proteins. So, first letter of Greek is alpha, so they mentioned alpha, so they put alpha helices. And the second one is the beta strands, right, and some strands they join together to form sheets. So, you can see the turns or bends, because if you have any regular secondary structures, helix or strand, if you want to change the direction, so it has some secondary structures which turns direction, right, that is mainly turns or you can call as bends, and some structures which are irregular. So, in this case you can call these structures as coil structures. So, you can have different secondary structures and the regular secondary structures are simplified to alpha helices and beta sheets along with turns and coils. So, how we get the helical structures? what is the interactions which maintain the alpha helical structures, right. You can see it is a kind of spring and the shape of a helix, right. You can see it is a shape of the helix. So, this is why we get the name helix, and the first letter of Greek is alpha. So, they call this as alpha helix. How alpha helix are formed? So, if you see the arrangement of residues, right, and the arrangement of atoms. So, you can see this one residue, it contains the atoms NH, CO, and you can see R is R group. What is R group? It is a side chain, right. How many side chains in a protein? 20 side chains, right. So, the R varies from 1 to 20. So, that depends. Then NHCO, this is the main chain, right. You can see this is the chain. Right, it will go like this. So, you can see C alpha, one side is hydrogen, one side this is R group. C here you can see the double bond O, N you can see the H. So, here you can see the carbon oxygen, and we have the amino hydrogen here, right. So, NH is here. So, between I and I plus 4th residues from the CO and NH groups, you can make the bonds. You can see from here this is NH and this is a CO, right. You can see the hydrogen bonds, this is hydrogen bond. You can see hydrogen bond. So, if you make these hydrogen bonds between I and I plus 4, between the NH and CO atoms, then automatically you can get a kind of spiral shape of structures. This is gives the formation of alpha helix. So, alpha helix is formed by hydrogen bonds between the CO and NH atoms of I and I plus 4th residues, right, in the main chain. So, if you look at the alpha helices, it is a kind of staircase just going from one to others means of some steps. If you go from starting from here and go here and then this will have one complete turn. If you go from here to here one complete turn, it contains 3.6 residues. For example, if you see this is 1, 2, 3 and 0.6, right. So, 3.6 residues per turn, and if you look at this complete turn, right, you can see this is the 5.4 angstrom in one turn, right. Now, 3.6 residues per turn and 5.4 angstrom per turn, then what is the rise per residue? 1.5, right. You can see rise, you can calculate 5.4 divided by 3.6, right, this equal to 1.5 angstrom, right. So, if you have for 10 residues, it will accommodate, if it is straight, then how much it will accommodate? 15 angstrom. This is the reason if you look into the transmembrane helices, the transmembrane segments they have up to 30 to 40 angstrom, right. If for example, if you take 30 angstrom, then how many amino acids can be resided in between the transmembrane segment? 20 residues, right. These are transmembrane segments are long, to accommodate the width of this 30 to 40 angstrom, right. You can get 20 to 30 residues, right, because there is not straight, it can have the different conformations. So, it can have accommodate more residues. So, approximately we get 25 to 26 residues inside the membrane. So, if you look into these helical regions, the residues are closely packed. For example, if you see these structures, right, they are very close, they are dense. So, they are closely packed in alpha helical structures, and there are several alpha helical segments in proteins, right, ranging from different residues. Approximately what is the average length of alpha helices in globular proteins? It is around 10 to 12 residues, right, approximately in known structures, approximately length of these alpha helices in 3D structures. So, this is another second one, right. First we discussed about alpha helix, and the second one is the beta strand, because it is a kind of ladder type of structures, right, and they find out second one. So, the Greek letter is beta. So, this put as the beta strand. So, there are two types of beta strands depending upon the direction of the chain. Here also you can see the beta strands which are formed between the NH and CO groups, right. You can see the CO group and NH group. You can see the hydrogen bonding between CO and NH to form the beta strand. So, here I give two different examples. Here this is the one which is here the chain goes in two different directions. One chain is going right to left and the second one is from left to right. In this case we call this beta sheet as the antiparallel beta sheet. And the second example, if you see the chain runs both from left to right the same direction. So, with the same direction then this is called parallel beta sheet. So, if we compare the alpha helices and beta sheets, alpha helices are closely packed and the beta sheets are completely extended and this is loosely packed. They can form the hydrogen bonds together to form the beta sheets either parallel or antiparallel depending on the direction. So, now we discussed about the alpha helices and beta strands, right. If you look into these structures other than the alpha helix, there are some other helices are also present depending upon the hydrogen bonding pattern. In the case of alpha helices, the hydrogen bond is in between I and I plus 4. Some cases you can see the hydrogen bond between I and I plus 3, right, and some cases you can see I and I plus 5. If the hydrogen bond is between I and I plus 3, right, then that is called, right, that is called 3-10 helix, right. So, there is 3 this is right. So, it is called a 3-10 helix, and if it is I and I plus 5, that is called pi helix. So, you have 3 different types of helices and which one is commonly occurring in protein structures? Alpha helix. Alpha helix is commonly occurring in the protein structures because it maintains the stability, right. Other if it is I and I plus 3 or I and I plus 5, it is either tightly packed or loosely packed. In the case of alpha helices, right, it's properly it's arranged. So, in this case it can maintain the stability and hence alpha helices are commonly occurring in protein structures. So, now we have other regular structures, right, called one is the turns. So, 1/3 of the all globular proteins, right, in all the among all the residues they contains turns because the turns will reverse the direction of the polypeptide chain. So, how turns are formed? So, what is the major interaction? This is also hydrogen bond, right, between the carbonyl oxygen, right, and the amide nitrogen between I and I plus 3. So, depending upon this the interactions, right, how they make the bonds, there are 3 different types of turns. For example, type 1, type 2 and type 3. And if you look into the frequency of occurrence of turns in protein structures, right, which one is most frequently occurring? Type 1, right. I will tell you what is type 1 and type 2 and type 3, and type 1 turns which are occurring more frequently than the other types, types 2 and 3. So, if you talk about the type 1 turns, it is mainly depending upon the backbone dihedral angles, right. So, you can see the residues are minus 60, minus 30 and minus 90, 0 for the residues I plus 1 and I plus 2 in the case of type 1 turn. And we look into the frequency of occurrence of amino acid residues in the turns, you can see the proline is the residue which presents quite often in the position I plus 1 in the type 1 turns because the phi angle is restricted to minus 60. So, I will discuss about the phi and psi angles, right, in a few minutes. Then in type 2 turns, in this case the dihedral angles are minus 60, 120, right, and 80, 0 for the case of I plus 1 and I plus 2. And here in this case the glycine is the favored residue for the case of type 2 turns. Here you can see the difference, the atoms here they change the directions of oxygens. So, this is called type 2 and type 2 dash, right. So, there is the negative and positive dihedrals. So, glycine is frequently occurring in type 2 turns. So, that is another turn called type 3 turns, right. It is not commonly occurring turns, but here you can see backbone dihedral angles are minus 60, minus 30 and minus 60, minus 30 for the case of I plus 1 and I plus 2 for the case of the class 3 turns. So, we discussed about different types of secondary structures. One is alpha helix, second beta strand and turn. All the secondary structures are formed by hydrogen bonds between the residues NH and CO in the main chain. So, now you can have a main chain, so write the formation of the main chain, right. So, you can see this starts from N C alpha C N C alpha C. So, we see the rotations at the different planes. You can see some places you can rotations are allowed and some cases rotations are not allowed. So, the formation of the dipeptides, that we have peptide 1 and peptide 1, how they are formed? The peptides by means of peptide bonds, right. For example, if you have the residue 1 and the residue 2, right, you can by elimination of water molecule, right, you can form the peptide bond. Peptide bond is the partial double bond character. So, it is a strong bond. For example, this is the peptide bond CN. Here the rotations are not possible. So, then if you look into these other bonds, the rotations are allowed in other places, right. What are the other two places? You can say N C alpha as well as C alpha C, right. For example, if you do this N C alpha C, N C alpha C, N C alpha C. So, here you can see Cn, this is the peptide bond. So, rotations are allowed, right. Here this is the peptide bond. Here you can carry the rotations, and here also you can see the rotations. Then the rotations along N C alpha, right, and this is called the phi angle, and the rotation along the C alpha C, that is called psi angle, right. If you write this phi and psi here, right, it is not a bond, it is a rotation around that particular bond. This is why they put kind of rotation like this, right, phi and psi. Right, how to get this phi and how to get this psi? Phi means the rotation along N C alpha, that means there are two planes, right, one form C N C alpha form one plane and N C alpha C forms another plane, and the rotation between those two planes, right, they form phi angles. So, how many atoms are required for the formation of this diagonal angles, either phi or psi? 4 atoms, right. So, which is the bond length? Okay, this will define bond length, right. How many atoms are required to define a bond length? 2 atoms, right. Then you can see angles. For example, you can see angle here, right. In this case, how many atoms are required to get an angle? 3 residues. So, if you guys 1, 2, 3, you can see an angle. But in the case of diagonal angle, how many atoms are required? 4 atoms are required. First 1, 2, 3 form a plane and 2, 3 form another plane. A diagonal angle is the angle between these two planes, right. Here in this case, we can see phi and psi angles. Now, the question is it allowed for any angle? Phi can take the rotation of how many degrees? 360 degrees. From we take start from 0, we can take the complete rotation, you can take 360 degrees, or if you take the clockwise and anti-clockwise, then plus 180 and minus 180. Likewise, psi you can see 360 degrees you can rotate, right. But if you rotate phi and psi angles, is it allowed that all rotations? It is not allowed. Why it is not allowed? Because it is surrounded with other atoms, right. If only this we have the only 4 atoms, then you can rotate. But in this case, you have the R groups and you have the hydrogen and you have other atoms. In this case, rotation is not allowed for all the rotations, right, restricted to some rotations. To understand the possibility of allowed rotations and the disallowed ones, G. N. Ramachandran, right, he is from India, he tried to construct models, right, by forming the small peptides, for example dipeptides, right. So, he tried to consider each atoms, right, as hot spheres, right. It has the vibrating this van der Waals radii, they consider the van der Waals radii, right, a small r and capital R based on these vibrations, right, and then he tried to rotate and see which rotations these atoms can interact or atoms are not forming any steric clash. For example, if there is capital R, this outer radius, right, 2 atoms, this is R A, this R B, right. For example, if you have 2 atoms, right. So, this is a small radius R A. So, this is R A. So, you can see here this is the small one is R B, right, and the this is R B. If the distance is more than this R A plus R B, they do not have any steric interactions. In this case, the rotations are possible, rotations are allowed. If it is less than R A plus R B and more than small r A plus small r B, then it is possible, but not freely possible. Right. In this case, they mentioned that it is partially allowed. When it is less than this small r A and small r B, this van der Waals radius they clash. In this case, it is not possible. This is called a disallowed region. So, he tried all the combinations, right, and then see which angles are allowed for different secondary structures. For example, if you have alpha helices, right, how the alpha helices are formed, just we discussed, right, I and I plus 4, right, between the NH and CO bonds, and they are like helical shape. In this case, the rotation is very restricted, because if you rotate more, automatically it will make a steric clash. So, in this case, they found that the phi angle of this is the range for phi angle and this is the range for psi angle, this is psi, this is phi, right. You put the x axis as phi, right, and the y axis as psi, right, and see this is the region where they can accommodate these residues, right, if it is an alpha helical conformation without any steric clashes. Here you can see the yellow one, is called the partially allowed, okay, this is the partially allowed regions, right, and the white region they are completely disallowed regions. So, compared to the alpha helices you can see the beta sheets, so you can see this is wide. Why it is wide?
Because more flexible.
More flexible, it is loosely packed, right, it is not tightly packed, it is loosely packed, so it can have more options for the interaction. So, you can see the more rotations, right, are allowed in the case of beta sheet. This way you can see it is mainly around the minus 180 regions and plus 180 regions. So, this is the phi and you can see this is psi, right, you can see. Then J. N. Ramachandran, right, so he got with these small models and then he checked with these known structures, right, and then see if we alpha helix structures we know. So, you can calculate the phi angle and psi angle and then we can verify whether the alpha phi angle and psi angle are within the limit what he proposed in this allowed region. So, in this case, okay, as we discussed, okay, these are the two bonds which are free to rotate. So, these rotating angles they call phi and psi. So, JNR use the computational models, right, and then based on that collisions he determined the allowed regions and disallowed regions, right, based on the sterically steric interactions. So, now how to calculate this phi angle and psi angle? For example, if you have protein 3D structures, I can give the coordinates, okay, this is the x, y, z coordinate. If you know the x, y, z coordinate, you can calculate the distance, right. How to calculate the distance?
Euclidean distance.
Euclidean distance, and Hamilton we discussed yesterday, x1 minus x to the whole square plus y1 minus y to the whole square plus z1 minus z to the whole square, right. Angles you can calculate, right, you can take the vectors and you can take the get the angles. Then how to get the torsional angles? If we have, for example, if we have this the x, y, z coordinates and take 4 atoms, right, here you have put the 4 atoms, this C, N, C alpha, C, I take the mentioned atoms. For the first one we know that x1, y1, z1, and the second one, for example, you take this as x2, y2, z2 and this one x3, y3, z3. If you have 3 coordinates, 3 atoms, can you make a equation of a plane? They can form a plane, right. What is the equation of a plane?
x1, x2.
Right. So, you can get the equation, right, a1x plus b1y plus d1z plus d1 equal to 0. So, a1, b1, c1 and d1 you can calculate using the determinants. Here 1, 1, 1, y1, y2, y3, z1, z2, z3 is a. Likewise, you can calculate b, c and d, right, because x, y, z all are known, right, because all the numbers are known, right, if the coordinates are known. If you do this, then you can form this equation, right, for plane 1. Then go for the plane 2, what other 3 residues you need to consider, right, we take this 1, 2, 3, then you take this as 1, this is 2 and take this as 3. Take these 3 coordinates and you can use the same equation, right, and then you can calculate the equation of the plane. So, that is equal to a2x plus b2y plus c2z plus d2 equal to 0. Now, you can calculate the diagonal angle using these 2 equations, right, using this cos alpha equal to a1 a2 plus b1 b2 plus b2 divided by square root of this. Now, if you get this one, then you can get the alpha, that is cos inverse of the quantity, that will give you the alpha, then you can get the diagonal angles. I show an example, the same data we have. So, there is a server which can take these x, y, z coordinates and get the equation of the plane. For example, if you have x, y, z coordinates, right, this is the same coordinate which I show here 9.176, okay, this is the coordinate for the first one, right. Okay, give then this is the equation of the plane. Then for the surface 2 you can get the equation of the plane, and finally with these 2 equations they calculate the diagonal angles, okay, this is about 77 degrees. So, if you get the coordinates of 4 atoms, because 4 atoms are required for diagonal angle. So, 3 in one plane and the 3 in another plane. So, you can get the equation of the planes and then use these equations to get the diagonal angles, right, it is very simple. Now, this is one example, for example, myoglobin you know this is all alpha protein, mainly alpha helices, and they showed that 60 to 69 these residues form alpha helix. How to verify, how to check whether these residues really form alpha helix or not?
Phi psi angle into.
So, get the phi psi angles. So, we take the mentioned atoms, get the phi psi angles and then see whether these phi psi angles are in the allowed regions of the Raman plot. So, if you get these 5 angles, that should be within this region. Within this region, then you can say that okay, these residues belong to this alpha helix, right? We can do that. So, now if you have a 3D structures based on the hydrogen bonding pattern, you can easily define secondary structures.